I'm going to switch gears a little bit and talk about some of our work that is focused on trying to use multi-omics to approach studying genetic diseases. I'll talk a little bit about modeling that we're doing that combines both sequence information and data that we're collecting from a wide range of individuals. But first, I want to set up the problem. I think many of you are probably aware that all the efforts to do whole genome sequencing have found rare variation at scale. This is abundant in human genomes. If you go back to got the taskbar open there. Doesn't matter. If you go from the 1,000 genomes where there is 2,500 people, you're seeing there's on the order of 50 million variants, all the way up to the recent version of All of Us, which is looking at 250,000 individuals now, where you have over 1 billion variants. If you imagine, again, the size of the genome only being 3 billion base pairs, we're effectively seeing an enormous amount of variation across the genome. And we might get to the point where all non-embryonic lethal variation has been surveyed or reported. This small number here is the UK Biobank. That's looking at close to 500,000 people or 450,000. The number is lower, but you're probably familiar. They only sequence exomes, so this is just the coding part of the genome. And if you go in and dig and look at this paper, what they actually report is that they discovered 12.3 million variants in 39 million base pairs, of which 99.6% were rare variants. So basically 1 every 4 bases, they're reporting a rare variant. Right? So this becomes an enormous interpretation challenge, and it's going to require the models that that Anshu was just chatting about before. And so the question that really drives a lot of work in the lab is, how do we actually understand the importance of all these rare non-coding variants, particularly given that we can't do things like statistical association, given that we see these things so few times. So there was a group that tried to figure out what would be the reporting criteria for a rare non-coding variant if we were going to use it in any clinical practice as part of medical genetics or clinical genetics. And they went about this problem thinking about, can we use the ACMG criterion effectively in the non-coding space for reporting benign, likely pathogenic variants in the same way we do for coding variants. And so I want to walk through just a little bit of this to set up the problem here, and particularly focusing in on the fact that to get to something that's strongly pathogenic, you need to have population data that shows that the variant is more prevalent in affected individuals compared to controls. When you look at computational and predictive data, you need to see the same predicted impact as an established pathogenic variant. Okay, so that's actually, I think I zoom in here, make it a little easier for reading. So that's actually not that interesting, right? You're basically saying I can call it pathogenic if I've seen it pathogenic before. To get to very strongly pathogenic, you get a predicted null variant in a gene where a loss of function effect is a known mechanism of the disease. This is only recommended for splice variants. So what this basically means is if we've established before that this has an effect and you're finding that same variant, well, you can call it strongly pathogenic. So really there aren't the computational and predictive tools that exist right now that clinical or medical geneticists would use in reporting something, that is strongly or potentially even moderately pathogenic back to a patient. So this is a major gap in how we report genetic information. And even with the best deep learning and AI tools, we're not there yet. We can't run Chrome BPNet in the clinic and then say, okay, we found this variant, and then that's good enough. So what's been required typically for this type of evidence to get to strong as well for non-coding is you need well-established quantitative functional studies in patient-derived tissues and cells that show a deleterious effect. So you can make the prediction, but you also need to do something where, in a cell line or from a patient sample, you show there's some kind of aberrant change for this particular variant. So I think the problem's easy then. We just have to go back and create cell lines for the billion variants that we've seen and observe all the different types of effects that will be observed, and then everything's solved. Unfortunately, that is a massively expensive and massively challenging problem to do. So one of the things that we've been trying to do is think about, is there evidence that sort of bridges between this, like things that we can get from individuals that we can combine with the types of advanced machine learning and statistical models to improve our characterization of important rare variants for an individual. And I'll talk about a few different stories here, but the way we've gone about this is recognizing that outlier phenotypes typically inform impactful rare variants in individuals. So if I can collect information about that person, in terms of phenotype, and say there's something that's really different than the population, there probably is, some increased prior for there being a genetic cause. This actually goes back maybe 15 years or so in the genetics literature. People used to sequence the outliers of a phenotypic distribution as potentially, being enriched for interesting rare variants. The way we've approached this is we can do this from molecular phenotypes. So we can survey things like gene expression or protein abundance, methylation in populations, and we can say, who are the people that carry these extreme values? Do they have interesting rare variants? And can we use this information together with sequence-based predictions or annotation-based predictions to help prioritize those as being important for that individual? Okay, so we report on this in different ways. One of our favorite studies to use for this is the GTEx study. So if you're not familiar with it, it's a multi-tissue map of gene expression across just over 800 individuals. There's about 40 tissues that were collected across the body, but the average number per person was around 20 or so. And we can use this data set to say for every individual, let's look at the expression of all of their genes and where it lies with respect to the rest of the individuals. And we can actually categorize what we call multi-tissue outliers as, every tissue we look in, this person has really aberrant expression, either over or under, compared to that population. And there are different molecular phenotypes we can extract from RNA-seq data that's present in GTEx. So we can obviously look at the simplest one at the bottom here, total expression, and try and identify people that are outliers. So this bottom here is showing just a measure of the Z-score and someone being highly overexpressed. We also get splicing information. So we can say, is someone using this junction in a particular way that we don't see it used that often compared to anyone else in the data set? And at the top, we can also look at allele-specific expression. So this is a property of sequencing data, whereas if you're sequencing over a heterozygous site, you can count the number of reads that are assigned to one allele versus the other, and you can look for extreme deviation from expectation that way as well. So we first asked, okay, for these molecular phenotypes, are rare variants enriched in these outliers across the allele-specific expression outliers, splicing outliers, and expression outliers? And we stratified this by allele frequency, going from low frequency, which is about 5 to 1%, rare, less than 1%, doubleton, singleton, novel variants. And we see for all these different categories of outliers that there were SNPs and indels that seem to be modestly enriched in some of these outliers. So this kind of sets up the prior that, okay, the extremes actually may have some interesting rare variants that are there. We then went a little bit deeper and we said, okay, which rare variants in these outliers tend to cause the largest effects? So it's a busy slide, but I'll walk you through it quickly. Actually, we'll just choose the splicing outlier one to walk through. So as you go down, we're increasing the strength of the outlier definition. So you can imagine increasing the Z-score threshold that we're finding things at. And we're saying what categories of different rare variants are we finding that are increasingly enriched under these stronger and stronger outliers. So for splicing outliers, it shouldn't be as a surprise that as we get to stronger and stronger outliers, we get more of this sort of blue color here. And these represent your splice region variants, your splice donor, splice acceptor variants. So the really strong splice outliers seem to be driven by canonical splice events. For underexpression outliers, we can go down here as well. We see when expression is really low compared to the population, we get this mass here, which are frame shift and stop gain variants. So these are likely inducing nonsense media decay and causing lower expression. But we also see the expected types of things like deletions, and for overexpression, we see duplications. So what's interesting about this is we can actually start, looking at individuals' personal omics data and saying, okay, if I, am scoring, or if I'm seeing this level of aberrant effect, I can actually have a prior as to what types of variants would drive that effect. I, for instance, don't see any of these large, underexpression outliers that seem to be driven by regulatory non-coding variants. Actually, a lot of them seem to be caused by structural variants and these nonsense-mediated decay variants. So my expectation is some of these big effect large variants, I now know where to look if I see this effect. I can sort of ignore maybe some of the other parts of the genome. Okay. We've also seen that as we collect these, we can start to think about how to integrate these with our understanding of genetic risk. Again, this is maybe a bit of a busy slide here, but some of you might be knowledgeable of polygenic risk scores, which try and summarize the additive contribution of genetics across the genome to explaining a particular trait. These are typically built from common variants. They don't include rare variants. And so we asked, if we make a prediction of a trait like BMI for an individual using a polygenic risk score, but we see their observed BMI that's measured in the UK Biobank deviates from that, do we see that there's any increased enrichment for them having these sort of strong effect outlier variants? And so what you're seeing at the bottom here is the deviation within the PRS group of the BMI score from expectation. And as we increase this deviation from expectation, they tend to have more and more of these sort of rare outlier variants that are present there. So it's giving us some clues that, okay, some of these variants might actually be interesting and impactful for complex traits. And if we could integrate them, we'd probably do a better job explaining genetic risk. Okay, so getting to the main thesis of the talk, we've been thinking about how we can actually combine information we're getting from DNA-based annotations and predictor models, like the ones Angela was talking about, with personal omics to prioritize rare variants. And so here, there's not a lot of models that combine all the data that you'll get from an individual. You might have sequence predictor models, which will, work from the DNA alone, but typically those don't actually take into account anything else that you could collect from that individual. So we work with the Battle Lab at Johns Hopkins Labs, as Alexis Battle's group, over the last few years to actually develop and iterate on a model that we called Watershed that allows us to integrate outlier signals and rare SNPs, and now recently structural variants to help prioritize interesting variants. And so the way this works is there's obviously two observed states here. So this is this node up here can be either for structural variants or single nucleotide variants, but it's basically a vector of genomic annotations for that particular SNP. So you're just saying, what is the genome annotation telling us? We also have outlier signals from a gene of interest for that individual. So that's the other observed state. We can, collect allele-specific expression outliers, splicing outliers, but this is something we observe there. And we train this model, so I keep going here. So the SV, we include different types of predictors, like whether or not it's an ABC enhancer, CAD sites, VEP scores, whatever we can get. And it's the same with SNPs. We can put in all sorts of different types of annotation there. And what we can try and do is train it to learn these unobserved variables of interest. And when we run this model subsequently, we're actually just trying to assess, what's the probability in that observed data of sort of matching these conditional probabilities here, and output from that a posterior that any given rare variant is likely to drive that combined or that observation of the extreme effect on expression given its genomic annotations as well. Okay. So this, as I mentioned, has been trained on N2 pair, or trained on observed GTEx data. Typically we do this on N2 pairs. It's as low into the allele frequency as we can get. So these are doubletons. We've observed a variant twice, and we were trying to see if we observe one with an outlier, how well do we do overall predicting the other one. So we've seen that this actually has large improvements over just the genomic annotation alone models. So this is just showing the precision recall curves for expression, allele-specific expression, and splicing rare variant prediction. So obviously, this is just showing how challenging this task is if you're just going in with like genome annotation information alone. This actually isn't including models like Splice AI or others, but it's very hard to predict these kinds of non-coding effects. We've also recently updated this for structural variants. And so where we think this is important is obviously, there's been enormous value to new approaches to do long read whole genome sequencing, but they're currently still reporting variants that we don't have large scale references for to understand their allele frequency. So we thought, well, if we can actually get multi-omics data for them, we can prioritize which SVs are most likely to be interesting to an individual in lieu of having any kind of population allele frequency. And so we've been able to compare this watershed model across different sequence contexts to the whole genome sequencing only model. Again, with precision recall, we're seeing in this blue line that we're doing a lot better. So this information is guiding us towards what we think are interesting rare variants where we can replicate molecular effects. So given we have this model, we've tried to assess, what is the potential disease relevance of these outlier variants beyond, sort of integrating them to polygenic risk. And what we were able to do in GTEx was actually just score like how many times we saw, or how many variants we saw per individual that had high watershed posteriors that were related to allele-specific expression outliers, splicing outliers, or just total expression outliers. And we saw on average, people had maybe about 5 to 7 or so interesting variants. So all of us in the room, each one of us has some interesting, transcriptome outliers, splicing outlier driving rare variants, which are not included in any genetic risk prediction currently. But some of these rare variants we're finding impact well-studied genes like ApoE, P53, et cetera. So we really want to push our ability to find these for individuals and then to understand their phenotypic impacts. So Anshu will mention this study, so I don't have to go into it too far, but we've been collecting data on non-European populations as well. And so one of the advantages of having models like this is that in other population studies where you may not have large sample sizes, you can go in and you can start to try and learn something about rare variants or low frequency variants, which might have functional effects where, the allele frequency could be 1% in a subpopulation in Africa and 0% everywhere else in the world. But that's, still carried by 1 in 50 individuals. So what we have done is we've run this on that cohort. We prioritize rare variants. I just want to give you one example of the kinds of things we're finding. This was just a splicing variant we found in the ELAC2 gene that replicated. This was from 7 individuals across these 5 different populations. So you can see the posterior here is really, really high. This variant is like, just under 1% in this cohort. It's not found anywhere else in NOMAD, right? So it's, and you can see there's just again, really large posterior for that. So we can actually go and look up this variant. And we find that this splicing variant is actually, it overlaps with a known GWAS loci, which from multi-ethnic studies of prostate cancer has exhibited large effect sizes. So in sort of this reverse genetics approach, we're finding these variants that seem to be correlated to interesting traits. Okay, so we've done this in a lot of contexts. I just want to use the last part of my talk. We've done this in populations, trios. I mentioned GTEx across tissues. We've looked in developmental contexts to see what kinds of rare variants we found. But I just want to talk about this last part, which we published in the last year or so, where we're starting to extend this model to look at outliers across different omes and integrating these together. And to do this, again, we're collaborating with the Battle Lab. We had access to data from the TopMed MESA cohort, which included whole genome data, but also a range of different omics from approximately 800 people measured at 2 different time points. So we actually get replication of some of these omics assays as well. This includes methylation, transcriptomics, and proteomics on top of the whole genome sequencing data. So we can do the same thing we did before. We can stratify, the effect or the magnitude of the outlier effects for methylation outliers, splicing, expression, protein. And we see that, again, there's like rare variant enrichments conditioned on these different outlier thresholds. We can also see different categories of variants are enriched again in all these different ohms. So this is just showing we see like splice, stop, frame shift, TSS, same kind of thing that I showed you earlier. But what's interesting is that we can actually start combining these pieces of data together, right? So we can say, what happens if we combine expression outliers and splicing outliers? Do we, so we see the same type of effect for the same type of gene in an individual. Does that increase our ability to find enrichments for interesting rare variants? In fact, it does. So this is this joint overlap, this orange dot here. So you can compare if I have something that's expression alone, or maybe splicing alone, I do see these rare variant enrichments. But when I see both the expression and the splicing effect at an outlier threshold, I'm starting to see even more enrichment of rare variants. This is true for expression in protein as well. So these orange dots here as well. So they tend to be increasingly enriched for stop gain and frame shift rare variants when you see that gene is both an outlier, for total expression and protein abundance. So we've gone farther. We've tried to build this multi-omics watershed model. Again, the same probabilistic graphical model framework as before, but now there's all of these different observations of omics for an individual. And the output of this is we can report posteriors about how many significant variants people have for each of the different ohms. And so, we're seeing on the order of this threshold of 0.5 in this data set, maybe closer to 60 or 70 or so expression rare variants, a large number impacting methylation, maybe about 20 or 30 impacting splicing, and a couple hundred impacting protein levels. So, I was mentioning we have a few, all of us have a few that impact expression, while we all have a few that impact a whole bunch of different types of ohms as well. So, and again, none of these are sort of systematically reported to any of us. We see these different ohms also prioritize different types of rare variants. So it's not like we're always getting the same thing if we do the proteome versus the transcriptome. And this is just a simple upset plot showing the intersection of the posteriors. So obviously lots for methylation here, but there's only about 26 high scoring rare variants where we see the impact across, all the different assays. Many of them are specific to a single assay or combinations of those assays. So I'm going to just leave on a few different points of how I think this type of data is useful and how we're starting to use it in human genetic settings. So obviously we can prioritize outlier rare variants with this model, and then we can go into GWAS, that have gone deeper into the allele frequency spectrum and say, do these variants that we prioritized have bigger impacts on traits? And so this is showing for different cutoffs here of different outlier effects. So this is your RNA, posterior 0.5, 0.9, methylation again, 0.5, 0.9, splicing, protein. As you get up to higher posteriors, where do those variants land in the effect size distribution for a polygenic trait like height? So this is looking at all variants for height. So millions of variants here make up this distribution. So they tend to be sort of approaching the top of those percentiles, right? And you see the same for splicing, same for protein, maybe a bigger distribution there. So we're able to prioritize things that are likely to have big impacts on traits. Many of them we don't know the biology of still. But we can also see when we combine these shared outlier signals, this goes up even further. So again, the height effect size percentile, if we combine things like splicing and methylation, well, we find the single top hit variant for height, right? You go straight to the top of those millions of variants, protein and methylation here. So these things are, they're giving us fewer variants, but I think they're giving us like the most meaningful large effect variants for some of these traits. Likewise, we can also flip the problem around. And so many of you are probably aware of like burden tests where you look for the repeated presence of rare variants in individuals with particular traits. So we went into the UK Biobank and we categorized people who had sort of a normal distribution of height versus an outlier height value. And we asked, well, for those people with outlier height values, how often do we see high scoring rare outlier variants in specific genes? And what we found through this approach is we were able to recapture some known Mendelian height genes like FBN1, CHSY, et cetera. So you can see the difference in the distribution of some of these high scoring watershed prioritized variants versus the total distribution here for each of these. Some of these are a little bit noisier distributions, but here's a clear difference as well. So again, we think this adds power to being able to do burden tests, and you can include variants outside of the protein coding genome by having this combined information. So just to leave with, what non-coding variants are important. Obviously, we want to get to the place where sequence-based models can do a great job in providing predictions of impactful variants that we can use in clinical settings. However, still, we're going to need functional systems that actually show the impact of those predictions. I think there's a future here for methods that integrate personal genome sequencing information with multi-omics, in all sorts of different frameworks. And not a lot of these models exist, so it's an opportunity for people to work on that will get us to the point where we can start calling important alleles in the non-coding genome, not only for research, but potentially in clinical settings as well. We all have these variants. Many of us don't know which ones they are. I don't know what mine are. But again, I think these integrative approaches are the things we need to be able to interpret new types of variation and move us forward. So with that, I'll just acknowledge my lab and my collaborators at the Hopkins lab, and I'm happy to take any questions.